All right, so let's talk about chromosomal mutations. Mutation, by simple definition, is any sort of mistake that happens either in the sequence of the chromosome itself, or in our case, we're going to focus more on big picture chromosomal mutations, so what happens when those chromosomes separate in meiosis or they go through crossing over and something goes wrong. Okay. What causes mutations? Some of this we know, some of this is still kind of coming to light. One of the things we know is that they can be caused by mutagens. Okay. So some sort of agent, a chemical, UV radiation, radioactive elements, something that ends up increasing the frequency in a mutation above and beyond sort of these spontaneous mutations we seem to know happen. Again, that word spontaneous probably just means we don't know why. Okay, we haven't figured it out yet. All right, something's gone wrong in your proofreading, which we'll talk about with your DNA coming up in the next unit, or, whatever. So mutations that occur in your gametes only, okay, so your sex cells, your egg and your sperm, are the only mutations you can pass along to an offspring. So let's just take the fact that UV light coming from the sun is a mutagen, all right? The biggest problem with that UV radiation is that it's coming in and it's hitting your skin cells. If you're not wearing proper protection, clothing, sunscreen, whatever, it can cause mutations in the division of your skin cells. What does that mean? Skin cancer. Okay? But you do not pass skin cancer onto your offspring. Why? Because you're not passing genetic information to your offspring via your skin cells. All right? You're not just sloshing off some skin cells and making a new baby. Okay? So in order for a mutation to get passed on to the next generation, to your offspring, those mutations have to happen in your sex cells. So for women, the mutation has to show up in an egg cell. For guys, it's got to show up in the sperm. All right? If those mutations do not happen in those cells, baby won't have the mutation. Okay? So you're not passing skin cancer on just because you weren't so good about wearing your sunscreen. You're not passing that on to the next generation. All right, it's going to affect you, but you only. Okay? Things like breast cancer, however, that show up in families, we know there's a genetic link to it. That mutation is showing up in the gametes, typically of women. Okay? So she is passing that genetic mutation for breast cancer potentially onto the next generation, all right, because it's showing up in those eggs. All right, so hopefully that difference makes sense, okay? Something else to point out, and we'll come back to talking about some other types of mutations next unit, but typically that word mutation has a connotation or has a meaning that people will assume is harmful. And you're right, most of the time, most mutations are not good. Okay? But sometimes they can be neutral, which means that mutation happens and we never really see any sort of manifestation of it. All right? Or there is a manifestation, you do see some sort of physical or behavioral change, but it doesn't really increase that organism's fitness, it doesn't decrease it either. Okay? So sort of thinking back to the evolution idea, what makes an organism more adapted to its environment? Well, one of the ways we get those different varieties is mutations. All right, a lot more of it's going to come from the crossing over and the independent assortment and the sexual reproduction we've been talking about with meiosis, but some of it can come from mutations. All right, and when they are beneficial, okay, so when they're good for the organism, they increase that organism's fitness, allow that organism to have more offspring than somebody without that trait, all of a sudden that becomes an increase in its fitness. Natural selection will probably select for it, and it becomes beneficial for the population as a whole as that mutation continues to become more prevalent, all right, or more frequent. Okay, so that's sort of mutations in a nutshell. we're going to focus on the chromosomal ones, so altering something within the chromosomes itself. One of the examples we already talked about, here's a karyotype, here's that trisomy 21, so here's 3 of the chromosomes instead of the 2 you would typically expect. This is Down syndrome, okay? Down syndrome is the result of something called non-disjunction, all right? So what does that mean? Non-disjunction happens during meiosis, okay, and it can happen in meiosis 1 or meiosis 2 when we go through anaphase, okay. What happens? The chromosomes sort of get sticky and they don't separate. So if we're talking about meiosis 1, rather than that homologous pair separating, that tetrad coming apart, all right, and one homologous pair going this way, the other going this way, they stick together, they get stuck. So you end up then going through normal meiosis 2, but this cell only got a quarter of the information, this cell got 3/4. Okay, so instead of being a nice 50-50 split, all right, we get some uneven split here. All right, what does that result with then? Okay, well, here are 2 cells that have an extra chromosome, and these 2 cells are lacking one. So each of these should have had 2 cells, or excuse me, 2 chromosomes in each cell. These 2 have 3, this one's got 1. So there's an unequal distribution of those sister chromatids at the end. So the information is doubled here, okay, and we're missing half the info here. It can also happen in meiosis 2. So we get through meiosis 1, normal tetrad split in anaphase 1, but when we get to anaphase 2, the sister chromatids in one of those cells don't come apart. So we get stickiness going on here. What does that mean? Well, we get 2 cells that are normal, so they each have 2 chromosomes, but when we go and divide this cell, this one's lacking a chromosome and this one's got extras. Okay, so that's the possibilities with that. We go through and get something that ends up with Down syndrome, all right, and one of these types of cells has been passed along, okay, so there's an extra set. Now, that's not the only chromosomal mutation. So non-disjunction literally means that that junction or that separation didn't happen. Okay, so it's not coming apart. That's what that means. Whenever you end up with extra chromosomes, okay, you got either more or you can have less, that's something they call aneuploidy. Okay, so somebody who has Down syndrome is an aneuploid organism, okay? So they've got an abnormal number of chromosomes. They've got 3 of 20, chromosome 21. In other cases, here's some of the sex chromosomes. I said sometimes you can add an extra X. All right, here's Klinefelter syndrome. You may have an extra Y. You may have 3 X's. You may be missing one. All right, here's the interesting thing, though, with the X's. All right, so this one's got 3 X's, but she doesn't actually have a abnormality. She doesn't have a syndrome. All right, we'll talk about when we get to genetics after spring break what happens to those extra X's. All right, having an extra Y, same deal, normal male. But you've got 2 X's and a Y. It's still male, but we got some extra female features in somebody with Klinefelter syndrome. All right, lacking that extra X, still a viable offspring, but there's also some abnormalities there. But she's female, there's no Y to kind of give that male characteristic to her. All right, so if you're interested in some of these, go for it. Do some research. It's some interesting stuff. But this is what's going on here. Okay, so frequency in the population, also you can see they're relatively rare. All right, but aneuploidy simply saying that there's an extra or less than the number of chromosomes that that person should have, okay, which is typically a result of non-disjunction. We can also have changes to the chromosome structure itself. So here's one chromosome, all right, here's another, And we can have these different possibilities. A lot of these alterations to an individual chromosome cause some sort of birth defect, or we see them showing up in cancers. Okay, so deletion simply means we take a section and now I've got less. Okay, so I should have had 4 sections here. Now I've only got 3. So I've deleted part of the information for that chromosome, so it's a little shorter than it should be. That's something they can also look for on a karyotype. All right, if they can't find matches, but the banding patterns seem to match, all right, we may end up with a shorter chromosome. You can also have a duplication. So here's your homologous chromosome. Typically, this is something that's happened during crossing over. Rather than it being an equal sharing of information that gets crossed over, one chromosome gets a little more than it's supposed to, all right, which will typically then lead to deletion on the other. So instead of having 4 sections, we now have 5. All right, and this stuff here in pink is the same information twice. Okay, so part of that chromosome got doubled. It should have gone to this chromosome, but instead it's got stuck here. The other option is some sort of inversion. Again, typically happening during crossing over, so it should have been pink-green, all right, when it crossed over, but something happened to that information as it crossed from, say, dad's information to mom's, and it got swapped, which means this whole set of genetic information, those DNA sequences, are backwards. That's a big problem because DNA can only get read in one direction. It's like you reading a textbook and I tell you, oh, read the sentence backwards. It all of a sudden doesn't make any sense. Okay? So the same thing happens here. That DNA can only get read in one direction. Now I'm basically putting the sentence backwards and it's not going to make sense to your body. Okay, so we're reversing the information. All right, so deletion, we remove a section, typically happening off of one of these chromosomes. The other one's probably getting a duplication, so double the information for some sort of gene, and then the inversion where we're swapping it, we're putting it in backwards. Okay. This one's an interesting one. So what they call reciprocal translocation. So what ends up happening is that 2 sections from non-homologous, okay, so not the same information, they're different chromosomes, say chromosome 1 and chromosome 2, swapped some information. Okay, so somehow either crossing over went wrong, that synapse at the very beginning to make those tetrads with the homologous chromosomes went wrong, and then crossing over happened after that. Or in some plants, this actually happens just normally. All right, but what they're doing is swapping completely different information. Okay, so crossing over goes between homologous chromosomes, so there should be 2 purples here, so that they're swapping the same info, all right, eye color for eye color or hair color for hair color. But now I'm swapping information, and this should be my trait for eye color should be here, but instead I've got information for height. Okay, and so the information that's getting swapped is on the wrong chromosomes, right? And that tends to cause a lot of problems as well. Okay.